Professor John Edmonds, who's Professor in the Centre for the Mathematical Modelling of Infectious Diseases at London School of Hygiene and Tropical.
The development and evaluation of new diagnostics, drugs and vaccines. Since the creation of regional clinical research networks around the world who are ready to respond to clinical research networks with the same solidarity that WHO encourages us to
protein into the body using the viral vector. Now, because the adenoviral vectors are...
So you must be working really hard at the moment.
Yes. Trying to do everything at the same time.
I think we're at a fairly critical stage just at the moment in being able to know what's going to happen. Are the number of cases being detected? We've heard that they're dropping. Is that because there are fewer infections really, or is it just difficulties in actually doing the tests and monitoring the patients? The next few weeks will be critical, and we can't at the moment say which way it's going to go.
About where you want to go next.
Okay, next I've got to go downstairs and next door to see somebody in the Wellcome Trust building.
At the moment we're doing lots of things in parallel. We are starting to test the vaccine in animals, so that will be happening over the next couple of months. We're also developing the plans for the clinical trial. We have to write a very large dossier of information to supply to the regulatory authorities to ask for approvals for the trial. We're helped in this by the fact that we've done very similar trials with very similar types of vaccines many times in the past. So all of that has to be prepared. The vaccine has to be manufactured in a very controlled condition in order for it to be ready for clinical testing.
Oh, right.
Yeah.
Okay.
In the case of healthcare workers, it might be to do with the exposure, because if people are getting a very high exposure from working in close contact with infectious patients, they're getting a lot of virus entering their body at the same time, and that's much more stress for the immune system to deal with. It makes it harder to control the infection, even in somebody who's young and otherwise healthy.
Vaccines. For some of the other technologies, DNA vaccines, you may need to give 2-3 doses.
Well, there are two main concerns, and they are always to do with what's not known. One is it's not known what the potential of this virus is. Could this virus actually become a permanent resident in humans and each year emerge again and cause disease, or is it a one-time event? WHO still feels it might be a one-time event and is recommending to do these rapid containment measures. And the other thing that's not understood is the seriousness of the disease. We've seen that it can be very serious in people with comorbidities, diabetes, lung disease, and in the elderly, and in a few of those who apparently are very healthy but young. So understanding the full spectrum of disease is very important as well.
Types of technologies are not so good for use in an outbreak, where you would really prefer to be able to...
We're really at the very early stages of developing a vaccine. We've had the advantage that the Chinese scientists shared the genetic sequence of the virus, and we've been able to identify the portion that encodes the surface protein of that virus, and we're using that sequence as a vaccine to inject into the muscle. It produces that same protein, it's only part of the virus, and evokes a protective immune response. We've gone from that sequence to a candidate in 14 days. We're starting animal testing, and the next phase will be to try and move it towards human testing. But that's a long process. It's going to take a while to check it's safe, and then see if it can block natural infection before it would be available for the global community.
Again, it's been modified to make it very safe, but it's a good way of getting the genetics.
But what we're really trying to position it is so that we're ready if this becomes a persistent problem on a global scale, or even just in China, that we can have a vaccine potentially ready by next year. Now that may be important if it becomes a seasonal event.
You talk about it in terms of a race and so on.
Work together on data collection tools to encourage standardised clinical data collection during outbreaks, preparedness and response, referred to as SRP.